Hey guys, welcome back. Last time we were here, we were familiarizing ourselves with genes, loci, and alleles. And today we're going to talk about some of Mendel's laws, how he figured those out, and how those apply today. So, with Mendel's law of segregations, we got homologous chromosomes and the alleles that they carry are going to segregate into separate gametes during meiosis. And so chromosome 1 goes one way, chromosome 1, the copy from the other parent, goes the other way. The plants that are homozygous for the dominant allele, big P, can donate a big P allele or a big P allele. So it doesn't matter, even though these two segregate during meiosis, the information they carry is the same. If you're homozygous recessive, you're going to split into little p or little p. And so all of the egg or sperm or pollen that you're making is only going to have little p. And then heterozygous plants can produce both types of gametes, the type of gamete that has a big P or a little p, depending on how these segregate. And so that's our first very important law. And so here you have big P, DNA replicates. Now you have two copies of it. Meiosis 1, we have two copies still. Meiosis 2, we have big P. Here you've done the exact same thing with little p. It's replicated. Meiosis 1, meiosis 2, little p. So then upon fertilization, we have big P and little p as our only two options. So these you could say were true breeding big P, true breeding little p, and what they made is what's called a heterozygote. When you see this big P, little p setup, that is heterozygous or a hybrid. So here's how DNA replication works. You guys know this from last chapter's meiosis, so you're going to be in good shape there. And 3 is, of course, fertilization. So probability. When you calculate probabilities, it's not that hard. You try to measure the chance that a particular outcome will occur. You've probably done this in math, and Punnett squares make this pretty easy to do. Punnett square is a grid that we use to calculate the probability of genotypes and phenotypes in our offspring. I'll show you Punnett squares for both a monohybrid cross in class, and we'll also do a dihybrid cross. And so Punnett squares are very useful. And here's how you do it. Let's say you have a male, and his genotype for a certain allele or a certain gene is big P. Those are the possible alleles he has. And you have a female that is big P, little p. So what are our options? What can we make out of this? What you do is you go ahead and carry this across. I think they might have made a mistake here. There's big P, little p. I think this should be a little p. What you do is you carry him across. Take the big P, and you write him in all the squares across. Take the little p, you write him in all the squares across. So that when you're done, you have big P, little p, big P, little p, big P, little p, big P, little p. And with a very simple Punnett square and a knowledge of the alleles the parents are carrying, we have determined the probability is 100% that they will produce hybrids, heterozygous hybrids, that will express the trait for purple flowers because they all have big P. So that's the phenotypic ratio is 100% purple flowers and 100% heterozygotes. That is our genotype ratio. And so once we do that, we can start to figure out some really cool stuff with what's called a test cross. A test cross is a method of determining if an individual is heterozygous or homozygous dominant. And so let's say you got a purple flower. This purple flower could be big P and be a purple flower, or he could be big P and be a purple flower. And we don't know which one he is. So what we do is we find a white flower because we know that white flower is little p and little p. And we cross him with the white flower. Now, if we cross him with the white flower and all of the offspring come out with purple flowers, we know he only donated big P, so he carried both of those alleles. If we cross him with a white flower and some of them come out with purple flowers and some of them come out with whites, we know that that original purple flowered plant was a heterozygote to begin with. And so we can figure that out with a test cross. So you want to cross them with a homozygous recessive, and you can figure out what you're working with. So a monohybrid cross is a little bit easier. It's a test cross that looks at two alleles at a single locus. And all you have to do is take two true breeding individuals, which is big P, little p, or identical heterozygotes, big P, and big P, and you do your Punnett square with those, and you figure out your probabilities with a monohybrid cross. And it's really very simple to do. We'll do that in class. So when you track your generations, because this is important to do, you need to track your generations with these numbers, or letters rather, P, F1, and F2. P is the parents, and F is the first filial generation. And so that's F1 and F2. And so basically P is the parents, F are their offspring, and F2 are their grandkids, if you will. So that's how you do this. So in a monohybrid test cross, your true breeding parents are crossed to get the F1 generation. As you cross the F1 generation, that's your actual test cross, and your F2 generation is what's going to give us these really nice ratios to figure out what alleles our F1 generation were carrying in the first place. So that was the bell. So I'm out of time. We'll draw these in class. They're really not too bad, and we'll practice these. So thanks for your time.